Hello, everyone. Welcome to webinar, seminar, and networking series on technologies for the future, revolutionizing drinking water safety in developing nations with AI-driven optics. For your kind information, this webinar is being recorded. If you have any queries, please type in the chat box, or if you really want to speak, please request in the chat box and we will enable your video. The main objective of the program is to identify the needs in developing countries, explore the solutions, and join hands and build collaborations. In our last webinar, we discussed about identifying the needs in developing countries identified by the stakeholders, and in this workshop, we will be discussing about identifying the needs and finding out the solutions. The program goes like this. The first speaker will be Ms. Purnika Thapa. She is an intern at Socialize NP, which is developing an AI-based platform, and she will be discussing about spatial and seasonal variations in water quality due to rapid urbanization. The next speaker will be Dr. Basanta Giri. He is a co-founder and a senior scientist at Kathmandu Institute of Applied Sciences. He will be discussing about smartphone microscopy and paper-based analytics for microplastic, oocyst, and fecal contaminations. The next speaker will be Professor Heidi Otavare, a professor at Faculty of Engineering of Free University. She will be discussing about Raman spectroscopy and static light scattering in microfluidic system. Then we'll have a discussion period, and after that, Mr. Alex Mawaki, Country Director of Safe Water and AIDS Project Kenya, will be presenting us about the problems of WASH regulations, implementations, auditing of water safety plans. The last speaker will be Professor Samuel Doravich, a professor at University of Illinois Chicago School of Public Health. He will be discussing about the production of ozone on-site using microplasma methods. Then we'll have a discussion after that. This webinar is organized by Futung Research Institute, supported by Royal Academy of Engineering, Optica Nepal section, and Research Institute for Bioscience and Biotechnology. Futung is a non-private research institute which was established in 2016 and focused on addressing ignored challenges in developing countries. And this is our teammate. Not only that, Futung does advanced research in nanophotonics, 3D imaging, water sensing, ultra-fast spectroscopy, light matter interaction, electronics, PIC-based Raman spectroscopy to address challenges of the developing countries. Now, I would like to ask Ms. Purnika Thapa to present her work. Thank you.
Good afternoon, everyone. Thank you for having me here. My name is Purnika Thapa, and I'm really excited to share with you some of my recent findings in the water quality here in the Kathmandu Valley. Now, millions of years ago, Kathmandu, the capital city of Nepal, once used to be a huge lake, which now is flat. There are some hills which were islands back then. Now, running across the valley from north to south, there are several rivers, largest of which being Bagmati. The name Bagmati, which stands for it descends from the surrounding hills. The scope of today's presentation is to discuss the physiochemical parameters, including pH, temperature, DO, BOD, COD, nitrate, phosphate, and ammonia. And these were the 8 sampling sites. The samples were collected from during dry and wet seasons. Now, talking about the study area, it was a 29-kilometer stretch of Bagmati River originating from Bagdwar to Chobar, with the elevation of Bagdwar being 2,690 meters to Chobar being 1,400 meters. The vegetation in the upper stretches, especially Bagdwar and Sundarijal, where dense forests were seen with pine, oak, and rhododendron. And as we move to the downstream, especially Chobar, Balkhu, the vegetation was sparse with more urban settlements and agricultural land. And this is a picture of Tirupurasar's temple in the dry season in the 1950s. And in the next, you can see the Tirupur is the same picture, but it's a more recent one of 2022 during wet season. You can see it's all flooded. So the samples were collected twice during wet season, which was in June and July of 2024, and dry season, which was February and March of 2023. The pH, temperature, and DO were analyzed on sites, but the Others were analyzed in the laboratory. So speaking about temperature, you can clearly see in the picture as we descend from 2,690 meters to 1,400 meters, the temperature quickly rises over a relatively short distance. In the wet season, it is much more lower. It is much more lower due to minimal solar insolation. Now, as we move to the pH ranged from 6.9 to 7.8 in the wet season, with higher pH seen at Bagdwar due to minimal human activity and and lower values seen at Chobar due to urban runoff and industrial discharge. As we move to the wet season, the pH decreased due to increased runoff carrying acidic substances. As we move to dissolved oxygen, the values were much more lower when compared to wet season due to reduced water flow and higher temperature. But in the wet season, it improved significantly due to cooler temperature and higher water flow. The BOD, the BOD showed quite consistent organic pollution. It showed that some sites were very high, but some were not. But the reason was unknown to us, despite the seasonal changes. And now moving to the COD, uh, the the values in dry season were much more were higher, but it was less when compared to wet season due to less dilution of pollution and low flow condition. In the wet seasons, the value elevated, which reflected higher pollution levels from runoff. For nitrate, in the dry season, it was higher, which reflected industrial and urban pollution. And in the wet season, it was lower when compared to dry season, which was due to dilution effect, though the nutrient runoff still continued. For phosphate, the values peaked at dry season, which was due to agriculture runoff and sewage discharge. The value of ammonia in the dry season were lower than the wet season, which reflected reduced microbial activities. And in the wet season, it was much more higher, which was influenced by increased microbial activity and pollution from agriculture and urban runoff. So in contrast between dry and wet season, the pH and temperature were slightly lower in the wet season, with high DO value in the wet season, which was due to cooler temperature and improved aeration. The BOD and COD were higher in the wet season, which was due to episodic pollution events from runoff, though BOD showed minimal seasonal variation. In conclusion, it showed that the water quality deteriorated significantly as it moved from its origin to its outflow during dry and wet seasons. The reasons include insolation, water volume, and seasonal human activities. Bagdwar showed good water quality, and Chobar showed the worst water quality, which significantly showed that the water quality deteriorated as we moved downstream due to urban and industrial areas. And in the dry season, the values were much more lower, which could be which could show further impact on aquatic life, public health, and ecosystem. So, and lastly, I would like to thank you for having me here today. If you have any questions, I'm free to answer.
So there seems to be no question in the chat box. I have got a question,
Yes, sure.
Purnika. In most of your slides where like you measured like nitrate and diesel oxygens and particularly what was it, BOD. So there is a deep in Pashupati area.
Yes.
Yes. So why do you think
For
it is?
BOD, like it's much more higher in Gokarneshwar than Pashupati area in the wet season. So the reason being, the sample that I collected from the Gokarneshwar was from a cremation site, and Pashupati I just collected it a little up where the residues from the dead bodies did not mix with the water. And also, another factor that might have affected could have been that while I was collecting the sample from Gokarneshwar, it was heavily raining. So maybe there could have been some dilution that caused my samples to be high in value.
So we did a similar analysis in the past using our TLF measurement system.
Mhm.
And what we found was, what we guessed was, because there is a water treatment site before Pashupati, because people take a dive in Pashupati area, right?
Mm.
Because it's considered a holy river. And there is upstream the Pashupati, there is this water treatment plant. Maybe that is a reason. I mean, just asking,
Mm.
how do you
Yeah, it could have been, but like you mentioned, the water treatment site was just a little up from Gaudikat, and you see the quality of water was significantly improved in Gaudikat because the water was just out from the treatment facilities.
Hmm. Hmm. Okay. I'd like to introduce what this program is about. So let me share my screen. So this is a series of webinars, seminars, and in-person seminars, online seminars, uh, to try to discuss about the problems in the developing countries and what could we do to address these challenges. What are the needs and what can we do to address those needs, and to try to find the solutions, to discuss the solutions. Today we will have a very good speakers, prominent speakers, researchers doing work in this area, and we will discuss those solutions, and then we will try to set up a collaboration network, you know. So that's the goal of this series, and it's partially funded by Optica Nepal section. It's a new section of Optica, previously called Optical Society of America in Nepal, and then Royal Academy of Engineering. So I was kind of named Distinguished International Associate from Royal Academy of Engineering, and to explore this kind of solutions. So I thought the best way is to discuss with the community, with the problem owners and the research community, and then make a collaboration between all the stakeholders. Right. So this is what it is, and just give you a brief overview of what. So we already have one webinar, and the second webinar we actually, because it was more about identifying need, we went to the people in the community and had we did not have the webinar, but we talked with them. But it's summary of those interviews and discussions are there already in the website, and more will come in the coming weeks. And there will be on November 21st, there will be an in-person workshop in Kathmandu. So if you have planned to, if you are not in Nepal, and maybe this could be a reason to be in Nepal, and there will be another in-person workshop in next year in April. And then there will be several webinars coming up where we will be discussing about, sharing the problems, finding the expertise, things like that. So this is in our menu in upcoming programs. And you can always ask questions to the speakers. You can type, right? I think this has already been told. So I think it's now time to go to next speaker. So we have Basant Giri, Dr. Basant Giri, who is senior scientist in Kathmandu, in KAIAS, Kathmandu Institute of Applied Sciences, and he's been working in water sector for a long time. He's applying artificial intelligence technologies and smart technologies I think his talk will be very important, or important in the sense the kind of technologies we will be seeking. So over to Basant. Thank you for joining in, and you can start your presentation.
So I'll be talking about our efforts to develop point of need technology for water quality testing at our institute, Kathmandu Institute of Applied Sciences. I'm working there as a senior scientist. My background is analytical chemistry. And thank you for inviting me to this webinar. So I'll be talking about three different works. The first one is what we call the micro-bio paper analytical device to detect E. coli in water samples. The second one will be a smartphone microscopic system to detect parasites in various samples, including water. And then the third one will be the detection of microplastics in water, also using a smartphone microscope. Before I jump into these three topics, I would like to take this opportunity to introduce our institute and then the work we do. Very brief. Kathmandu Institute of Applied Sciences is a not-for-profit research organization in Nepal, established 10 years ago. We are about 30 researchers. Primarily, we do research, but we also do outreach activities. And there are three different centers active in this organization. For example, Center for Analytical Sciences, I belong to this center. Center for Water and Atmospheric Research, and Center for Conservation Biology. Typically, so this is the numbers from last year in 2023. So this was what we do in this institute. Primarily, we do research, so publish research papers, give presentations, organize some events, scientific events. And the work we do ranges from developing point of need technology to food and drug quality testing, environmental pollutions, including both air quality and water quality. Our colleagues also do remote sensing to look at the drought, extreme weathers, climate change, wildlife, biodiversity, sustainable development, all these things. And we also, as a part of the outreach activities, we organize seminars, workshops, conferences, and we also provide short trainings to early career researchers. And collaboration is important. So we collaborate with a lot of other researchers from across the globe. As you can see in this slide. And these are the partner organizations, mostly funding agencies and project partners. So my research group at CAIAS focuses on 4 different areas. The first one is the development of point of need technology. And I also do food and drug quality screening work, environmental pollution monitoring, and a little bit of science policy, science diplomacy, lobbying for good policy in Nepal. So our approach is we look at the local problems faced by our people in their everyday life, but also make sure that these problems we want to tackle have global significance, important in other countries as well. And one of the strategies for our group is to develop point of need technology to solve those problems. Okay, let me start with the water quality. The previous speaker highlighted the quality of water in our Bagmati River. And if you translate that into human health, globally, around 600 million children lack safely managed drinking water. That's a big number. And a lot of people die, millions of people die actually. So about 1,000 children under 5 years old die every day, which is very unfortunate because they do not have safe drinking water. I want to give you why the point of need technology is important in our parts of the world by giving an example. So this is a photograph from one of the incidents in Kapilvastu municipality in Kapilvastu district of Nepal in 2021. There was a diarrheal outbreak, about 1,000 cases and few deaths. But for many days, we were not sure what caused the diarrheal outbreak. That's because the local hospitals, local laboratories were not able to detect the pathogens because they did not have the required resources on site at their facility and the cost associated with this. So in this kind of scenario, here comes the importance of point of need technology, which are low cost, easy to use, and they may not need sophisticated laboratory facility and highly skilled human resources. There are plenty of methods to detect various contaminants in water and, for example, also in food. They are good, well established. They produce accurate and reliable data. However, most of these equipments are not portable. They are very expensive for us, and they require a skilled technician to operate and are not appropriate for field settings, for example, in emergency settings. And so we would like to, therefore, we would like to work on the point of need technology. This technology refers to the tools and systems designed to provide immediate access to services, information, or diagnostic capabilities exactly when and where they are needed, often without the need for centralized infrastructure. They are portable, user-friendly, and should give results in less time. So these kind of technologies are ideal for use in remote locations. For example, our country, in emergency situations, for example, we get earthquake every now and then, or places that are lacking advanced laboratory facilities. Very good examples: the blood glucose monitor, glucometer, to test the blood sugar in diabetes patients. And we have seen COVID-19 rapid tests. And these point of need technologies actually fulfill the World Health Organization's assured criteria. And that means affordable, cost-wise. They should be sensitive, specific, user-friendly, rapid and robust, equipment-free, and delivered. So these point of need technologies fulfill these WHO's assured criteria. And our approach has been for past 10 years, combining paper analytical device, or what we call the paper microfluidics, with smartphone. So we typically do essays on paper device and then use smartphone to read the signal, analyze the signal, and report the result to the users. And lately, we have been also integrating artificial intelligence for more reliable data. So let's briefly talk about the microbiopad I was talking about. So the microbiopad we developed for detecting E. coli is about the size of the your business card, visiting card, and it is made up of filter paper. And it has various samples loading zone. This is the latest design you see on the left of your screen. And it utilizes very little sample solutions, around 5 microliter. And when you put the sample solution with other reagents, it gives, and after incubation for some time, it gives a result. If positive sample, you get blue color. And we are not inventing a new science here. This is already established science or chemistry. where the galactosidase produced by the E. coli, it converts the X-gal into a dye, and that dye is a indigo dye, which has a distinct color. So by identifying whether you get a color or not, or measuring the intensity of the color, you can tell whether the sample has E. coli or not. And we developed the device, and then we tested the device in water samples collected from different parts of Nepal, including Kathmandu, Chitwan, and also Pokhara, but we did not include the data from Pokhara in this paper. Drinking water, river water, pond water, different types of water samples. And if you look at the table, and so we compared the data from our microbiopad with the established routine method, as what is known as the MPN method. Very good correlation between these two methods, both in drinking water samples and also in river water samples. So in brief, the microbiopad requires less time of incubation. It is 100% accurate when compared to MPN method, provided the E. coli in the sample is more than 3 CFU per ml. And what we found is 93% samples were in Kathmandu were contaminated with E. coli, less contamination in Chitwan, and then the number of E. coli found was higher, obviously, in Kathmandu samples than in Chitwan samples. So from these results, what we found is this microbiopad could be useful at this current stage as a relatively faster semi-quantitative, semi-quantitative device to determine the E. coli contamination. As you know, E. coli is some sort of proxy as a fecal contamination of water. But it still requires, because our long-term goal is to have such devices in the field, in remote locations where there is no maybe electricity, no lab facility. But the current setup, current device still requires incubation device, so which may not be available in the field conditions. It still requires field validation. So we collected samples and tested in the lab for this work, but the device needs validation in the field, and then the further study will focus on optimizing other parameters so that we can detect lower concentrations of E. coli. So that's all about the E. coli and microbiopad. Let me move to the smartphone microscope. So this is the brief summary of the smartphone microscope that we have developed. It's basically physically modifying the smartphone back camera with a ball lens, as you see in the upper left corner here. And then we also have optimized the imaging stage for this microscope, its lighting conditions and other parameters. And with this setup, so this was published a few years ago, and only spending $15 US, except the microscope, except the smartphone, you can have a low-cost smartphone or expensive smartphone. But if you spend $15 US to modify the smartphone, the microscope stage, you can have this smartphone microscope. On the top right corner, you see the microscope being used, and then the figure in the image on the middle is the image captured by this microscope for Giardia and Cryptosporidium. Remember, the Giardia and Cryptosporidium are the major causes of diarrhea in developing countries. These are tiny parasites, cysts. They range from 5 to 10 micron in dimension. And this microscope was good enough to detect, to differentiate these two parasites. We also compared this microscope with the commercial bright field microscope and also fluorescence microscope, and we got comparable results. And because this technology was interesting, and then the water quality issue is very pertinent here, many Nepali media picked up our research paper and highlighted our work in their publications. And this work was done in collaboration with Dr. Bhanu Neupani from Tribhuvan University. So we have used this microscope to test thousands of vegetable samples collected from different parts of Nepal, and then the many samples were contaminated with some samples with both GRD and cryptosporidium, and some with only GRD and others with only cryptosporidium. So the smartphone microscope I just explained requires a person to take an image of the sample. And by looking at the image by human eyes, you need to identify whether the object seen in the image is Giardia or not. And also, if Giardia and cryptosporidium are present in the image, somebody has to count them, how many of them are there. And that's tedious when we are talking about hundreds or thousands of samples, and you will have many images per sample. So we are talking about large number of images, and that's tedious work. You need a skilled human resource. You also so there will be some human error when humans are doing these detections. And then we thought about developing an automated system to detect these parasites in the images. So in this work, I collaborate with Dr. Bishesh Khanal from NAMI Research Institute here in Kathmandu. And in our preliminary work just published a few months ago, highlights the deep learning assisted detection and quantification of cysts and oocysts on smartphone microscopic images. And we tested 3 different types of models: RCNN, RetinaNet, and YOLOB. We also employed 2 non-expert humans. So they are the students who were not exposed, who did not have experience of using the microscope or identifying the cysts in the images. And we had a human expert who has experience of a couple of years working on the same project. That's the ground truth. And we found that some of these models are pretty good, but not very highly accurate. Partly, that time we did not have large number of data set, because these models require large number of data sets for training. And then we wrote a proposal and got funding from Laguna Fund. And in this project, what we call the AI-assisted microscopy, using smartphone microscopes for detection of parasites. Our goal is to build a comprehensive data set of annotated microscopic images containing Giardia and cryptosporidium cysts in 3 different types of samples. Water samples. Water samples means all type of water samples. For example, drinking water that may come from a municipally supplied coming from pipes, or hand pumps, pond water, lake water, river water, all sorts of water samples. Vegetable samples. Vegetables we target mainly those that are consumed less cooked or not uncooked. For example, salads. Also stool samples. So we have collaborated with 3 different hospitals, one in the eastern part of Nepal, one in Madhya province, and one in the Sudur Paschim province of Nepal. And so we have thousands of these samples, and then hundreds of thousands of images. To be exact, I think we have around 500,000 images to train and evaluate the AI models. So we have not completed this project yet, but I'm excited to see the results. That's all about the smartphone microscope. Let me talk a little bit about the new work we have started in collaboration with Dr. Bhanu Neupani from Tribhuvan University. And this is about the microplastics. You know, plastic materials, and they are everywhere, from our households to, outside from your house. They are tiny particles, typically smaller than 5 millimeter. But they are found in all sorts of places. For example, they look like something like this. Mostly the fibers in the D you see here, and some are pellets type, some are fragments. And when you look at the scientific literature, this is a very growing field. What is called the emerging contaminant. So the number of research papers is exponentially going up in last decade. And especially the people from environmental science sector are doing more research on microplastics. Also from agricultural field, from chemistry and material science, from medicine, a lot of people are now focusing on microplastics. So these microplastics, the health impact of microplastics is not fully known, but they have mechanical inflammatory responses. They are harmful to humans by both physical and chemical pathways and cause problems in marine organisms and aquatic lives. And they also have ecological effects of microplastics, a lot of bad effect on the organisms and other ecosystems. And they have been detected in oceans, river, water and sediment both. You name the place. For example, in Mount Everest, people have detected microplastics. In Arctic, in soil, plant tissue, human tissue as well. So, for example, human brains, blood, and fetus, you name it. Mostly found microplastics are polypropylene, polystyrene kind of materials. I'm not going to in details about those, but just would like to give you a brief overview. So we have done little work and published few papers already. For example, the first one is how the Guheswari water treatment plant, that's what the previous speaker was talking about, just above the Pasupati Nath temple in Bagmati River. There is one wastewater treatment plant, and we looked at how that wastewater treatment plant is able to remove microplastics from river water or wastewater. And recently, we published one paper that describes the smartphone microscopic method. So we have a smartphone microscope, and we use that to look at the microplastic particles in drinking water. Just to summarize that paper, around 73 particles per liter were found in drinking water we measured, even though the number of samples, the focus of this paper was to develop a new method, but we also tested some samples, both bottled water and jar water. Bottled water is typically a 1 liter water bottle, and jar water is typically, I think, a 20-liter water jar, which is reused. And interestingly, we found that the number of particles in bottled water is less than the number of particles in jar water. Remember, we only were able to look at the particles that are bigger than 50 micron. So there may be more than these particles in drinking water, which we did not, were not able to measure. And one thing I would like to share here that some of these works are, people are concerned about the quality of our water. And when we published our paper, a lot of Nepali media picked up this work, and some online social media groups also picked up. And this shows the interest of public and how they are concerned about the quality of water and food. If you like to know more about our work on microplastics, there is a conference on 4th and 5th October this year in Hyatt Palace Hotel in Kathmandu. The name of the conference is the International Conference on Mountains in Changing World, and we are organizing a special symposium on microplastics. And so these are the presentations, the work from our group on microplastics. If you are interested, you all are invited to join and listen our work. With that, I would like to conclude my talk, and then a few take-home messages from my talk is water quality is a serious problem in Nepal, and people are very concerned about the quality of especially the drinking water, also the river water. But research in water quality testing, especially when you are talking about the affordable technology, is neglected. But there is a need for affordable and portable technology for testing water quality. And because this webinar is also for networking and collaboration, I would like to emphasize the collaboration is needed among researchers, practitioners, and policymakers from both low and middle income countries and developed countries to develop affordable tools to test water quality, which will obviously positively impact the lives of millions of people which are dying every day. Thank you so much. So the work I explained in this talk were carried out in collaboration with Dr. Bises Khanal from NAMI Institute, Dr. Bhanu Neupane from Tribhuvan University, and Dr. Reena Khadka from California State University, and funded by the Lacuna Fund, USAID, and the National Science Foundation in the US, and all my students who carried out this work. Thank you so much, and I would be happy to take any questions you have.
What a awesome work you just talked about. I think it's very pertinent to what we want to discuss about. And I think the next speaker, Professor Hedy, will also probably touch upon some microplastic research she is doing. And yes, and she will also kind of connect it to how North and South can collaborate probably, right? So let's, there are a couple of questions for you. So the first one is from Anis, who says, in microbiopad for E. coli detection, what are the reagents? How costly are they? And then there is a follow-up on that by Regine, who says, how about its self-life? And about incubation, how costly it would be, and if there are counting because of the incubation issues. And also probably what concentration of E. coli, probably what the question could mean is how much, because you are measuring in microliters. What if, because WHO says you have to measure in 100 ml of water, right? So how do you deal with this? I think you can summarize all these questions in one thing.
Okay, thank you for the questions. I think these are very important questions. Regarding the question by Anis, so what we did was we just used a different platform and used the same reaction people are using every day. So in terms of the reagent cost, the same reagent, for example, X-gal, but we use in a small amount, so that would save cost. Okay. The other question was how much time does it take to obtain the result? So the most of the time is for the incubation, and we have shown in this paper that for some samples, 8-hour incubation is enough, but that's not very distinct. So we set a 24-hour incubation. So that's the time limit for the result. And about Rajin Pradhan's question on the self-life of the pad. So we still have to work on that because we did not take the pad outside the lab. we put together the reagents and did the sample testing. So we have not done that test. That's very important factor for these kind of point of need technologies that can be used in the field conditions. And the question from Asim Maharjan. So this is question about incubation. So again, I think I mentioned a little bit, but still we did not replace the conventional incubation. So we shortened the time for incubation, but incubator is required. But maybe in future, somebody will come up with electricity-free portable small incubator that can be used for a small paper device. And the other question about the sample volume, I think we used 100 microliter of sample. And the question is how representative is that the sample adequate? That's important question. I do not have answer for that now. We try to have smaller volume of reagents and samples, considering you are testing water samples in remote locations. Maybe it's not easy to get large volume of samples. So we considered those factors and try to have the device with lower volume of samples. The other question is for AI-assisted microscopy, what type of data? So the data here is image, microscopic images.
I think the question was probably you have any database that you use. I think.
No, the data set is created by ourselves.
Oh,
so
yes, okay.
all the images were captured by our group.
Right.
All right. Thank you so much.
Well, thank you very much, Dr. Basant. And hope that you will continue engage with this community and make this community grow larger. Right. So let me introduce briefly Professor Hedy Otevara. Professor Hedy, like I had a privilege to be actually student of hers long time back, not very long time back, when I was Erasmus Mundus scholar in Belgium. So she is a very great teacher. So I have a very lot of influence from her. And she is very much, in my impression, as I remember her, she is very much industry oriented, practical oriented. I think that's what I still remember. And she is engaged in a lot of, these practical laboratory works and stuff. So really privileged to have her here, and we'll be listening to her talk. So it's all yours now, Eddie.
Okay, thank you, Ashim, for this very nice introduction. It's also my pleasure to be with you. So you can hear me? And you also see my slides. Perfect. Okay, so indeed, as Ashim said, I'm a professor at the University of Brussels. And during this presentation today, I will give you a flavor of the work we do with respect to optics and photonics to have cleaner water. You will see throughout my presentation that we also work on other topics, but in view of time, of course, I decided to focus on water. That is also the context or the context of your symposium. And the previous speaker gave already a very good introduction, because my main focus will be on microplastics. And the reason why I chose microplastics is because it's both a topic of interest to people and the community in Western countries as well as in developing countries. So, and this might be an ideal opportunity indeed to collaborate, as Ashim told us earlier. So, but I mentioned already to you that we will use optics and photonics. Photonics is the science and technology of light that will help us to innovate. And especially with the unique properties of light, we can do that. And during my presentation, I will mainly focus on what we do with optics and photonics towards the detection of different contaminants in water. Now, before starting, I just introduce this to you because it's a very important slide. It's the 8 key emerging technologies that were defined by the European Commission some years ago. So these are the 8 key technologies on which Europe will focus the coming decades. And you see that photonics is one of them, but next to that, there is also biotechnology, which is of course very important. Nanotechnology, AI is very important. And the nice thing about detection and screening of water is that it's a very multidisciplinary topic. And as such, you need to combine different of these technologies together. And that's also where Europe currently is focusing on. If we submit projects to the European Commission, they want us to be multidisciplinary. And I think that's the first message I want to give you. And what is also already as an outcome of the previous presentations, if you want to solve problems, you have to team up with people from different disciplines. That is very important to be successful. Now, we at the university in Brussels, we are what we call the Brussels Photonics team. We are a group of 80 researchers, and we are working on different topics in optics and photonics. But we all follow the workflow, which you can see here. Because once you have a certain problem, it is very important to first determine the specifications and the requirements for your specific application. For example, what type of material might you need for your sensor? Because if you take the case of microplastics, you will not make your sensor out of plastic, for example. But for some other applications, you might need biodegradable materials. So therefore, it's very important to choose the right materials. And for that, we collaborate with a group at Ghent University, which is another university here in Belgium. And with that group, we develop custom materials that might help us for some of our applications. We also have in-house and commercial available optical modeling tools that allow us to determine the required specifications of optical system which we develop. One of the important things there is if you then in the end want to realize and fabricate your system is that you have a tolerant system. What do we mean by that? It's a system which is not very sensitive to how you assemble this. And it's very important what we heard already in the previous presentation as well. It's very important to have a low cost, handheld and portable system. Of course, this will be used by non-experts, non-technic or not photonic skilled people. And for them, it's very important that it's a robust system or what we call a tolerant system, which is not sensitive to how you align things. And that is very important if you want to develop a system that you all take those tolerances into account. Next, we have at our facilities also mastering and prototyping technologies. I will show you those later. We also have a clean room with all optical measurement and characterization tools. If required for some companies, they want to develop some sensors and they want to send out those sensors to some of their customers to do first check. It's important that we also can deliver low volume replication of optical components. And in the end, of course, demonstrating and prototyping your final sensor is, of course, of crucial importance. So before now going towards the application, I want to show you first some of the aspects in this workflow, which might be of interest to you to know that this is available. So we have a pilot line for advanced prototyping of both plastic and glass optics. And this can be from regular optics, spherical, but also free form, any shape of optics, towards microfluidic channels and so on. So on the top picture on the right hand side, you see their ultra precision diamond tooling, which allows us to go towards very accurate surfaces down to micro scale, such that also, for example, diffractive optical components can be fabricated. On the left-hand side, you see some replication technologies like injection molding machines, which allows us then to do this replication. At the bottom, you see polishing machines. These are important when we work in glass, because there, with milling, your surface finish cannot be with optical surface quality. So therefore, also polishing is required. So of course, if you fabricate optics, microfluidics, or any optomechanical component, it is very important that you can characterize those components. And that is the reason why we also have a clean room with lots of metrology. And there we have different interferometers, profilers, microscopes that allow us to characterize the geometrical and optical properties of all the components we fabricate. For example, this can be, if you, for example, make a microfluidic channel, this might be the width, the height, the surface finish at the bottom, and so on. So if you think of lenses, of course, you want that your lens is performing well without giving any aberrations, blurring effects, and so on. So we can also measure those aberrations or optical properties. So, and this information, of course, is coupled back to our technology experts operating the machines, and they can then further optimize the manufacturing process. At the top right here, you also see some spectroscopic systems, and I will come back later to those, because those two are confocal microscopes and spectrometer systems. So this is a fluorescence confocal microscope and a Raman confocal system as well. So I will come back to that later in my presentation, as we will use them in our research. And of course, then you come to the prototyping, where you have to develop the system, assemble everything. And of course, as we work with different type of material, it goes from microplastics, but we also work a lot with biological material. We also have the biolab with all the required equipment for working with cells, proteins, microorganisms, and so on. So these are the facilities we have to our we have available in our lab. There are more which I couldn't focus on, but later on I will show you or give you a link to a virtual visit of our labs, and there you will see much more of our equipment. So this equipment is available for all the researchers in our group, and of course also to all collaborators with whom we work together. And if we then look to our group, we have 5 different sub-teams. And some of you might know the word TRL here at the left-hand side. It's a very important word which is used a lot in Europe. It stands for Technology Readiness Level. And it means if you're at the TRL 1 to 4, you're doing really fundamental research with its main result to obtain papers and some breakthroughs at fundamental level. Once you start to build real prototypes and demonstrators that can be tested by companies or institutes in the field, in their environment, for example, here what we heard in the previous presentation, close to a lake or a water pool, then we go towards a higher TRL, which is TRL 6. And if you end up in the end with a commercial product, you're at even a higher TRL 8, 9, and so on. So we have 2 groups within our Brussels Photonics team that work on fundamental aspects. One is on semiconductor lasers, where they look into novel properties of those lasers. And a second group is on photonic integrated chips, mainly there on graphene-based, diamond-based application of the pics in order to create special aspects. But I won't focus on those. I will focus on what we see at the bottom. So we have freeform optics. So they develop any non-rotational symmetric optics to have both imaging and illumination in various applications. Then we have a group working on optical fibers, not for telecommunication, but for sensing applications. And they embed those specialty optical fibers in different materials like composite materials for measuring strain, cracks, and do structural health monitoring, as they say. What I will present to you today is the work we do in the group on biophotonics and spectroscopy, of which I'm the head of. So our main goal is that we develop low-cost, portable systems that can measure faster with a minimal volume, and that you bring as much as possible also laboratory things like sample preparation onto the chip. For that, we collaborate with the microfluidics group at our university, because they allow to also have chromatographic columns on chip, which allows us to do also separation on chip. The main goal is to have this chip and not put it under a large and bulky microscope, but to have it in a handheld system, which you even could insert your disposable chip into your handheld system and make the readout as required. Of course, if you do a readout, it's very important that you choose the optical detection technique very well. And when light interacts with material, it can behave in different ways. So, of course, we can check the fluorescence. Fluorescence, as you all know, it's a molecule selective and sensitive technique. However, there is one thing you have to take into account. You need labeling. Except, of course, if you work with autofluorescent material, but labeling is required, and this might, for some applications, be asking too much or even might induce toxic effects. Therefore, people go towards UV-vis absorption, and there no labeling is required, but you have a very short interaction length of your light with your molecule under test. And due to Lambert's-Beer law, this interaction length immediately defines the sensitivity you have. And that is, of course, something which is very important to take into account, because in a microfluidic chip, you want everything very compact, distances are small, which would induce that your interaction length is very small and that you would have a low sensitivity. Therefore, it's important to find smart ways to enhance your interaction length when you work with UV-vis. Also, scattering might be very important, and especially in the case of particles and microplastic particles, it allows us to determine the size and the concentration of those particles. We also work on Raman spectroscopy, where no labeling is required, and as you can see here on the picture at the bottom left, it is based on energy shifts between the incident laser power, which is here shown in green, and the scattered light here shown in red and blue. And the fingerprint, which is very specific to the chemical compound that you measure, can be used to identify or even to track chemical changes. Now, Raman spectroscopy is very interesting, but it has a very low Raman scattering intensity. And as such, you need a very strong effect before it can be measured. And especially when you go to low-cost systems, this might be a problem. Therefore, people more and more work towards enhancement processes, and one of them is surface-enhanced Raman spectroscopy. How can you now achieve that? By using metallic nanostructures. This can be nanostructuring a surface, or you could even use metallic nanoparticles. Because these structures or particles, they absorb the incoming energy, And the electric field here at the top in this bottom right graph releases it locally into the tight gaps here between the two particles, and there, or it can be around sharp edges and so on, but those local fields here we call the hotspots. And then when chemical balance gets into these hotspots, the chance of Raman scattering increases, and thus, in other words, the Raman signal is enhanced, and that's what we call surface enhanced Raman scattering. So now that I explained to you the different techniques, the different techniques, I want to show you a bit the flow line we followed over the years. So I started the group in 2006, and at that moment we were making very simple systems for both fluorescence and absorption detection, and that went with, for example, capillaries through which your sample was going through, and then the optical detection was done around the capillary. Some of them we also had the microfluidics rotated by 90 degrees into the plane, as you could see here, and then you end up with special systems. We did that for various applications, to, for example, zebrafish embryo characterization, to even lubricant oil monitoring to check the quality of oil. More and more, we brought microfluidics into the system. We also started to work with biodegradable material, because in Western countries, of course, we have the possibility to do recycling, although this not always happens. But when you work then with polymers, it is very important that you also can, first of all, go to green polymers, as we call them, and also to polymers that might be biodegrade. And at that time, we worked on a project for the detection of malaria. There was a project together with some African partners where we did, based upon surface plasma resonance, we did this detection. Also, the microfluidics was added, which allowed us to also do a lot of sample preparation on chip. Then later on, we added Raman spectroscopy, as well as in the beginning optical trapping. But later on, you will see also in my examples later on that we also use other ways of trapping and not only optical trapping. We look to enhancing through surface enhanced Raman scattering substrates. And then we started more and more also to focus on very specific applications where it was crucial to have very sensitive detection techniques to have an early diagnosis, or what we call early monitoring. So, and this we do with partners from Europe, but also from all over the world. We have a chair together with Hamamatsu. You all might know Hamamatsu as a Japanese company selling a lot of optics and detectors. So for a few years, we have a chair together with them, and biophotonics is one of the tracks on which we work together with them. So here in my next slide, you see the applications on which we are currently working. So I will just highlight some of them, and then I will focus on the microplastics in water. So when developing new drugs, of course, it is very important that they also can be tested not only on animals, but also on humans. And with the whole ethical question about the use of animals for drug testing, people more and more look towards organ on chip. And we look to liver on chip because liver is the organ which is most sensitive to toxic products. So what we develop is a microfluidic platform together with a readout system, such that we can check the impact of toxic products like drugs, for example, on the liver cells by monitoring different markers like lipids, for example, collagen, and so forth. We also have a few European-funded projects on diabetes monitoring. Today, when people suffer from diabetes, Typically, one of the hemoglobin variants is measured. However, there are a lot of false positives, and especially for certain regions in the world where people do have a different hemoglobin matrix than others, it is important to have a full picture of the full hemoglobin matrix within your body. And therefore, we're developing a microfluidic chip, which where you enter a drop of blood. It is prepared through chromatography into different compounds, and then we do a Raman measuring combined with UV vis monitoring. So I said already, multidisciplinarity is very important, but more and more you also see that multifunctional or multimodal testing is also important. And what do I mean with that? That people combine various techniques, like, for example, absorption and Raman spectroscopy together to have a much better ID and characterization platform than just looking to one specific detection type. We also work on bacteria and especially antibiotic susceptibility testing. That's one of the big problems also in Europe, that doctors give too much antibiotics and we are becoming resistant towards different kinds of bacteria. So therefore it's important to be able to measure in a very rapid way if people are infected by bacteria, and in case yes, what kind of bacteria they are infected with, and then to check what type of antibiotics works and with which concentration, such that your dose is not way too high as it often happens today. And then we also have some research projects on neurodegenerative diseases like Alzheimer and ALS. But as I said already, I will focus on microplastics. If you want more information, you can look on our website or contact me for this. Now, the previous speaker already introduced to us that microplastics is a big issue in Nepal and surrounding countries. Well, it is the same in Europe. So if you look to analyses that are made, we see that about 3.5 million particles reach us only by drinking water. And then let's not forget what other microplastic particles are present in the food we eat, in the air we breathe, and as such, even they find microplastics in unborn babies. That's the reason why UNESCO said that as one of the targets, the important thing there is to have clean water. And therefore, within the framework of a doctoral training network, which was supported by the European Commission, we set up a project to characterize those microplastics. And we've used Raman spectroscopy and light scattering, and then first with bulky instrumentation, and then bringing it to an optofluidic chip that allows you to do this detection on chip. Here, I think I can go very fast, as this was already presented by the previous speaker. Microplastics, they come from different sources, they exist out of different materials, and we have them in different sizes. And what we see nowadays is that if we want to analyze these microplastic particles, it is very important that we can determine their number, shape, size, composition, and the source from where they come. Today, there exists bulky and very expensive equipment, which you can see here. They all have their pros and cons and the limitation with respect to the smallest particles they can measure. But what is very important is that we focus on measurement techniques that can go very small. Small, I mean below 50 micron particles and lower. And that's not, for example, possible with visual inspection techniques. And if we look to FTIR or mass spectroscopy, they cannot measure all the properties we are interested in. So, but why is it now important to go into the small regimes? Because studies have shown that if you look to microplastics in bottled water, and that was the focus on the European project I'm presenting to you, 95% of the detected microplastics are smaller than 5 micron. So this means that our focus should really go to these microplastics. Yeah. And we have been investigating various detection techniques. One of them is light scattering. What do we use there? Well, we use what we call a reflectometer. In fact, it's a very simple tool where we use a source, here indicated by L, and then we have in half a sphere a detection arm that can rotate in the plane as well as according to a certain angle. And within that half sphere, we have a detector, which is here indicated by PD, a photodiode, and we can move the detector on a mechanical arm in half a sphere. It can be a photodiode, but it can also be a spectrometer. In our study, we've been looking to the 3 most important polymers that we find nowadays in Europe in water, and these are polystyrene, PMMA, and polyethylene, and we also use them as standard references. We've looked to uniform and non-uniform size distributions. We call uniform monodisperse and non-uniform polydisperse. And what we did is we put there a cuvette with microplastic particles, and we sent light into the particles. Of course, part of the light is transmitted and part of the light is scattered. Now, if you look to that scattered light, we first of all see that the calibration of this light and of the whole technique is size dependent. And you can already see here, if we look to the scattered light, we see that some curves are overlapping. So therefore, if we want to do a quantitative analysis, it is very important that we first can determine the size. And in order to measure the size, we will use chemometrics. Chemometrics is a simple technique to, first of all, reduce the number of markers through principal component analysis. And then we use linear discriminant analysis to determine the size. And later on, once the size is known, we can determine the concentration. And what have we done? We have done this chemometrics and we have classified different particles, and we saw that for particles up to 1 micron, we can do very good classification. Going down below 1 micron induces some additional errors. So, and that's the reason why we have been focusing on particles from around 1 to 20 micron, which are also very important when looking to water contamination. Now, suppose we now know the size of these microplastics, it is very important then to determine out of a linear fit how the concentration can be determined. So we did that for both monodisperse, but also for polydisperse samples. So what we did is we mixed up particles, but prior to doing that, we divided them in 3 groups, the ones which we classified as small, medium, and large, and we mixed them up in various ways. We also did those scattering measurements, and here at the bottom right from this graph you can see, for example, the bluish dots are 2 micron, the red ones are 3 micron, and when you have mixed samples or samples with mixed particles from 2 and 3, you see that you nicely end up between 2 and 3 micron. The same holds for the other particles. So this allowed us to, first of all, determine in which class of particles they were. And once that was known, then again, we could determine the concentration by going into a linear fit. This is all further detailed in a publication which we published some time ago. Now, it's very good to determine the size and the concentration of plastics, but in most cases, people from the field, as we also could hear in the previous presentation, are also interested in composition and to perform the identification. And for that, we use Raman spectroscopy. Now, we also see that if you use Raman spectroscopy and you take a fingerprint, for example, you can here see the fingerprint of polystyrene and polyethylene, and you can nicely see that you can discriminate it. First, we used our bulky Raman system, and we did both point measurements. So we made a map by measuring several points, but we also did line scans, line per line, to increase the speed of measurement. And what we could see here is that if we look into a mix of polypropylene and low-density polyethylene in a ratio of 75/25, after Raman mapping, just in 2D, different points, we could see that our accuracy was not so good, because we found out that there was 54% polypropylene, while in fact there was 75%. And the same for polyethylene. We also found around 50%, but there was only 25%. So therefore, we see that it is required not only to look to 2D map, but also to a 3D map if you have what we call blended polymers. And that's what you nowadays have more and more. If particles starts to aggregate, different materials aggregate, and they form these blended polymers. That's why we also looked at to 3D mapping. Here we made a mix of 50/50 PP and polyethylene. And again, we did the Raman map. And here you could see that we came much closer to what was required, which is, of course, very important. Now, in order to quantify how good you do the mapping and how accurate you do the mapping, we defined a term which we call concentration estimation error, which is in fact the ratio between the real concentration of your polymer and the estimated concentration of your polymer. And if you then compare here on the top right, the concentration estimation error for both 2D and 3D mapping, you see that for 3D mapping, the error is much lower. Of course, there is one big disadvantage, that is the measurement time. Just for your information, to capture such a 3D map, it took 56 hours. That's way too long. So therefore, we switched to laser line focus, and there we will not work point by point, but we scan per lines. And if we do that, we could reduce the measurement time by almost a factor of 30. So we could reduce it to 2 hours. And you see that the error is in the same range as the 3D mapping. So this gives us a way to reduce the measurement time for more for analytical labs that are working on this polymer identification. Of course, that's not yet what can be used in the field. However, if we want to bring it to the field, we have to make everything more compact, and that's something we do through a microfluidic sensor. And of course, it's very important that we can keep the sample there for a small while, especially if you do Raman spectroscopy, it takes some time. And if you have a very fast flow, it's of course very important to be able to keep your sample there for a while. That's the reason why, if we would do that, we calculated that this is not high throughput at all, because to measure 1 micron plastic particles, you could only send 72 nanoliters per hour. So, and therefore, we developed a chip which we made out of glass, of course, because we are looking to polymer, and we use some acoustics. Acoustics, for that, we, of course, need to know the speed of the sounding fluid, the resonance frequency we choose, and out of that, you can determine the channel width or your chamber width. We fabricated the chip, we did some simulations, and in the end, you could see here is a chamber full with particles. If you switch on the acousto-optics, then you could see that all the particles are getting into a line. This makes it, of course, much easier to do Raman. Now, before closing my presentation, I want to show you also that, of course, now we could get all the particles within a line, but you also should be able then to collect them in a very good way. Now, if you would just think of a microchannel, rays, would just not all rays, depending upon the properties, could leave the chip. That's the reason why we made a chip with some half ball lenses together with a collector lens in order to capture much more of that scattered light. So you have here a spherical reflector at the bottom, and then some ball lenses and a collection lens to capture the light. We fabricated those chips. So here you see the reflector, and on top of that, we position the microfluidic chip with the ball lenses, and here you find the actuator. We measured the signal both without optics, just with the acoustics to get everything in line, and then with the optics to capture as much light. And with the optics, we could enhance the sensitivity by a factor of 32%. And you can also see it here from the number of counts you get. So also this was published in a paper, which you can look up later on. So as a conclusion, I want to say you that we have a pilot line that allows us to do optical modeling, design, metrology, optical detection, and this all with the goal. In the framework of my group, we make miniaturized sensitive platforms, but of course, this goes much broader for various applications where we combine different optical detection techniques with always the message early monitoring, early diagnosis, and that is very important nowadays. And finally, I would like to thank all the members of my group who helped to have this all or make all this true. And this is our website, bfot.org. And here you can find a virtual visit of our lab in case you're interested to scroll through. And with this, I would like to thank you for listening to my presentation, and I'm happy to answer any questions. These are also the papers of my PhD students who worked on these topics and which you can consult in case you're interested.
Great. Thanks a lot, Heidi. So it's very pertinent to what we are discussing. So the title or the theme of our whole series is Technologies for the Future. So that is actually very good question relating to this. So it's partly also my question addressed to me, but I would like to let you say why it is important for developing countries, why photonics and AI, the theme of this talk is important for the developing countries. I think I'll let you start.
That's a very good question, Hashim. Well, if you look back to developing countries, there you have special needs. And the thing is that in developing countries, you might have rural areas where people do not have all the labs with fancy equipment, as I have shown you here, what we have in our clean room. So therefore, it's very important to offer to people in developing countries ways to improve their life, and especially to improve the quality of their life. And that goes in different ways. It can be with respect to the climate, but with respect to the food, with respect to their health. And of course, all this is only possible with new technology. Over the years, many years, lots of things have been developed, but often it's either fundamental, people publish it in papers and they put it in the cupboard and then they hope that someone might pick it up. But we see also in Europe that there is an evolution to also bring it towards the people who need it. And therefore, I think, especially for developing countries, it's very important we look to more applied research. Of course, it all starts with fundamental aspects, and you don't hear me saying that we don't need fundamental research. On the contrary, we need it. It's the first step towards what we really want to bring the technology to the people. And the most important reason is to improve their life in all ways. And I mentioned a few examples, but it also goes to improve the education for children, to give more opportunities to people and students. And that's also for the people present here, why we also have scholarships in our master program in order to give those opportunities for people, because we need technical people. We need people engineers, physicists, biological oriented people, whatever technology they are interested in, we need these people to reach that goal.
Yeah.
That's my opinion.
Yes, let me add a few things. So a lot of new research is happening in developed countries, and this is the technological shift that is happening. So, so if we in developing countries are not aware of this technology and do not engage in this kind of research, then we will still be left behind. That's one reason, additional reason than what you mentioned. And this second reason, I think, is also these technologies are scalable. You know, the AI technologies, the photonics technologies, you can manufacturing a lot of chips that you are developing. When it's fully developed, we can manufacture a lot. And then the cost per
True.
device will
Just
be
for
huge.
the people to know, I think everyone knows Lego bricks.
Yeah.
Lego bricks are the example of mass manufacturing through injection molding. These are plastic chips, they are injection molded and they can be produced in millions of copies. Indeed, that's a nice example. And that's what you also can do with every photonic or electronic chip in the end. Yeah.
Right. So now let's go to some technical questions. So here, another Ashim is there who asks, when you have sample with microplastics, the plastics ought to move around the sample. Have you observed any signal fluctuations that can be attributed to those fluctuations? And if so, how long is the duration and how significant is the fluctuation?
Okay, but that highly depends upon the measurement technique you use, because for some measurements where you just take a snap and you can measure in a microsecond, you take a frame, that's not of a problem. So, like for example, for scattering, it goes if you just take a full field with a few cameras, that goes very fast. That's not of a problem. If you think to other techniques like Raman, it is very important. It's a crucial point. And that's why at the end of my presentation, I went a bit fast there because I was lack of time. But that's why we use acoustics. Because with acoustics, you can bring, as I showed you, you can bring all the particles to a place where you are interested in. And for example, here, so you can bring all the particles in a line and they stay there as long as you put the right frequency and you keep the frequency on, the particles stay there. If you release the frequency, again, they will be mixed up wherever and they will move. So this is one way in order to keep the particles on track if you want to then measure during a longer time than, for example, the gravity required. And as such, you can, this is acoustic trapping, but you could also go to other trapping techniques. There are even ways, what I've recently seen is that people also use mechanical traps that might be even more robust in the field. Mechanical traps to keep particles there, you can measure them and then you know the result. So there are various ways, but it highly depends upon your detection technique and the speed of your detection technique.
So probably connected to that, the question is, so in Raman spectroscopy, how do you determine the size, the distribution of the size?
Well,
And
there the
last
goal is
question.
that we now extend the chip which I showed you here with Raman, that we add imaging functionalities. Imaging functionalities or even the scattering, what I showed you before, that you have, because now we did a scattering measurement over a full half sphere, but of course we don't need all the angles. So you could, for example, foresee 4 or 5 angles, out of which you take a measurement, and out of that you determine the size and the concentration of the particles. And that comes again to what I told, multimodal sensing, combining different optical techniques in order to have the full picture of your sample under test.
Right, there are more questions, but we will move on because we are a bit, late. So,
But people can always contact me. So whatever you're interested in, also for collaborations, of course, we are open for that.
sure, sure. Now, let me introduce our next speaker. So far we've been very technical, mixed, but let's talk about the problem and the experience of Mr. Alex Malky, who is a country director of Safe Water and AIDS Project, SWAP, in Kenya. So it's very important for us to listen to the experiences in context of water, water problems beyond in Asia, which we know a bit, and Europe and the US. But also let's listen to Mr. Alex. And he is country director. I think he has a vast experience, 31 years of experience working in the wash sector, and wash is dear to him, as he says. So, Alex, so do you have a presentation? So I have already made you co-host.
Oh,
If you
well,
have, right.
oh, well,
You can-
thank you, Ashim.
Thank you very much for coming in.
I did do a presentation because I knew this was going to be a discussion,
Okay.
and so I will just be giving experiences on issues of water regulations, implementation, and even the auditing of water safety plans.
Yes.
As you rightly put it, my name is Alex. I'm based in Western Kenya. That is at the lake shore of Lake Victoria, at the shores of Lake Victoria. And I've been implementing WASH activities, water sanitation and hygiene for close to 31 years, as you've indicated. I work with the Safe Water and AIDS project, and in Safe Water and AIDS project, we try to come up with innovative solutions for improved health and economic status of the communities. WASH still remains a big challenge, especially in our country.
Alex, just a quick one. So
Yes.
can you turn on your camera so that people, otherwise, if it's not required, but if you can, otherwise it's fine.
Yeah. So speaking about WASH in Kenya, and in particular Western Kenya, we still have many households who still don't have access to safe drinking water, including sanitation, and hygiene is also compromised. As a country, we have regulations governing water. I remember in 2010 when we brought in the new constitution in the country, we the national government was kind of devolved into 47 subnationals, and in these 47 subnationals, water, health became a devolved function, and therefore this was now meant for county governments to ensure how can they improve access to water, issues of sanitation and hygiene. And so the national government was now left to develop policies and provide advisory services, including policy direction, in the event that we need to review the acts, like we have the Water Act, then that is the duty, responsibility of the national government. But then counties were required to ensure that they do allocate resources that will improve water situation, especially in marginalized areas. This includes the low-income areas, peri-urban, and even the rural communities, because those are the areas that are worst hit. In our Water Act of 2016, there was the establishment of the Water Resource Water Regulatory Service Board, which was mandated to ensure the rights and access to safe drinking water. Of course, this is not one sector operation, but we have multi-sectoral agencies which come together to address issues of WASH including our own Ministry of Health and through the Kenya Environmental Sanitation and Hygiene Policy. That again gives strategies for counties on how to ensure that those without access to safe drinking water, then how do we ensure that these problems are addressed? We've had quite a number of innovations, technologies that have been put in place to ensure that we meet the Sustainable Development Goals in Kenya. We also have our Vision 2030, and in line with our constitution, how do we ensure that every Kenyan have access to safe drinking water, including sanitation as a right? So, but then we experience challenges with implementation. For instance, issues of financial gaps. We still have problems when it comes to resource allocation, where you find limited or budgetary allocations are made for accessing water. And some of these technologies sometimes are very expensive. For instance, if you are to provide drilling of boreholes, unless you have external support, then you might find it's very hard or difficult for even county governments to allocate these resources. Then we also have issues of data integrity, like even trying to determine users or trying to ensure that we getting closer to accessing coverage of water to the marginalized communities. I recall 2 years back, we also decided to work with our collaborators to test protected water sources. And in one of the counties in Western Kenya, we did some water quality monitoring close to 97 protected water sources. And in all those 97 water sources, only 2 met the WHO standards and even Kenyan standards for safe drinking water. Those were sources that didn't have E. coli detection in the water. So that's triggered a lot, especially in terms of how can we strengthen issues of water quality monitoring. And I'm glad from the previous speakers, there are new technologies that would even be useful, especially in detection of E. coli in water. Over the years, I know this is a challenge across the globe, and in relation to climate resilience, and as a country, we have also observed situations where we have extreme drought, then we have extreme heavy rains that causes floods. This has also brought about challenges with access to adequate water supply, including sanitation and hygiene. And that has even resulted to outbreaks, disease outbreaks such as cholera and other wash-related infections. But again, we appreciate the fact that working with partners at the regional, at the national, and even at the global level, including research institution, we have tried to come up with technologies that can address issues of access to water, irrespective of the sources of water which people are using. And I'm sure the next presenter will be speaking about some of the technologies like solar ozonation that we have adopted. And this is also addressing issues of climate change. I know at the regulatory body at the national level, we've had also challenges with inadequate collaboration, especially with the national agencies. You will note that in this country, like we have different agencies, like in the water sector, at the national level, we have the regulatory body, we have the Water Resource Authority, we have the Rainwater Harvesting Authority. But again, there's no collaboration, especially between these agencies, on how do we address issues of access to water and sanitation services. Of course, it's supposed to be complementary between the agencies and even the ministries at the national and even at the county level. But again, I recall 2 decades ago, we were also trying to look at what is it that we can do even at household level to ensure that people have access to safe drinking water. And I remember very well in 2000, the year 2000, we started point of use water treatment, as has been also mentioned here by previous speakers. And so as a country, we've tried to come up with techniques or technologies that are adaptive at household level that they can use to ensure that they have access to safe drinking water, and not necessarily in absence of emergency situations like floods where families are evacuated and living in evacuation camps. But even in those emergency situations, how do we ensure that there's point of use access to safe drinking water? Of course, sanitation still remains a challenge. Hygiene also remains a challenge. But again, we have tried to accompany those with social behavior change, communication, and then we have introduced also market-based sanitation, where we tend to ensure that even with the low cost or local ways of waste management, you can still be able to prevent spread of diseases that could be attributed to maybe poor sanitation. And so those are some of the areas that we have also been trying to address. I know one of the other things, as I said, issues of water quality monitoring and even surveillance, that remains still a challenge. We are not doing very well, and even facilities that are supposed to be supporting these initiatives, maybe they lack funding, they lack human resource to do the work. But then, like for us as an organization in the region, we have also tried to extend that service where we test water and provide people with results on what is the status of their drinking water. I know as a country, we also try to have impact measures, trying to monitor the various indicators for WASH. For instance, the Water Services Regulatory Board do every 2 years, they conduct impact assessment of the water utilities and even water service providers, and they look at sanitation across the country. We have the Kenya Water Sanitation Civil Society Network, and also they do reporting from the non-state actors on what progress have they made in terms of accessing the vulnerable community lacking access to water, sanitation, and hygiene. And even they look at investments. What investments have we set aside to ensure that we have access to safe drinking water? So yes, we do have very good policies, strategies, but again, a big challenge remains is how do we operationalize these policies? How do we make them effective? And even now, as I speak, because they have been water and health is default function, then can those policies be customized at the county levels? And how can we ensure that there's effective dispensation these bills and even strategies that accompany the same. So yeah, basically a lot also needs to happen. And as an organization, we have been involved in a lot of research work areas in addressing some of these pertinent issues. So if you visit our website, we have shared some of those researches on technologies, innovations for WASH that we have been involved, and where people can see what else can be tried elsewhere, where we still have gaps in relation to WASH services. So thank you so much for listening to me, and I'm sure there could be questions on the same.
Yeah, so I have very couple of questions regarding your experience in Kenya. So one of the things is things that I am personally kind of thinking about is so far all the problems, all the standards has been developed, keeping I think from the evidence that is gathered in the developed countries. For example, okay, there has to be only like less than 1 CFU per 100 ml, right? For example, for E. coli. There are many WHO standards that is being put into put into effect based on evidence that was probably gathered in the developed country and uh, less so in developing countries. So, in your opinion, do you think that the current indicators, current standards, they need to be contextualized to the local needs? For example, in Nepal, the standards could probably be changed, should probably be, because, for example, the gold standard is plate counting, and then you have to have the whole facility, laboratory facilities and everything. So do you think, or in your opinion, do you think that this has to be updated, or because it's based on evidence, so it has to be left as it is now? So what do you think about it?
Yeah, thank you, Ashim, and that's a very good question. Yeah, so in Kenya, as much as we all use the WHO guidelines, especially in issues to do with safe drinking water standards, here in our country, we also have Kenya Bureau of Standards that is also giving us what standards do we need to meet, especially in terms of water quality. And therefore, those are the ones that we use. And even for our water quality laboratory, we give results based on both Kenya and WHO standards. So what are we finding out when we do this testing? So yeah, we have also adopted some of those standards locally as a nation.
Okay, so in our context, so in Nepal, so most of these standards, the local standards, are actually bit lowered version of the WHO standard, or most of the times it's the copy of WHO standard. So it's not well researched. So do you think there is some research need into like adopting this into local context? For example, E. coli by itself is most of the times is not bad for humans. E. coli, it doesn't cause you diarrhea right away because it is an indicator of fecal contamination. But like some people could say, okay, E. coli is not a problem. Let's try to look into typhoid, or cholera, So, but nobody has done this research. So I'm just blabbering, like giving my thoughts there. But do you think that there needs to be such research within our countries to set a different standard, or is it in Kenya you're happy with whatever the national standard is?
Okay, yeah. So our national standards in Kenya are also very close to what's WHO standards. But again, as you rightly put it, that of course, E. coli is a detection of contamination of water, but we have not gone into situations where we try to isolate whether we have problem with salmonella, maybe contamination in water, or amoeba in water. But again, this is an area that as an organization, we have worked even with local and even international institutions, and even trying to venture into wastewater-based epidemiological study to isolate what kind of pathogens are we likely to detect in the water. And that has been quite useful for us to know what type of bacteria are we likely to encounter, and is it maybe what is attributed to, for instance, when we have cholera outbreak. To an extent that even we have done recently even testing for malaria DNA in wastewater. So then we can be able to detect whether, yes, this is the probable cause of why we are having so many cases of malaria and not. Yes.
Right. So one question here from Om Vishnu, who asked, in your experience, what approaches have been most effective in helping vulnerable communities maintain access to clean water and better hygiene? How can we empower these communities to sustain these efforts in the long term? It's very policy related, and maybe you are the perfect person to answer this question.
Very good, very good. Yeah, thank you for that question. Yeah, so for instance, for SWAP, we work with very vulnerable or marginalized communities who don't have access to safe drinking water. And one of the areas that we have invested a lot is through the community strategy. Like in this country, we have community strategies, and the first level of service delivery, which is level 1, begins with the community health promoters. So we have worked very closely with these community health promoters who actually move from door to door, and they're able to promote issues of primary health care at the household level. And they, during their door-to-door campaigns in the households, they're able to demonstrate how to make your water safe using various methods, technologies. For instance, use of sodium hypochlorite solution, which is locally available even in those remote areas. There are other technologies that they can also apply. For instance, use of the aqua tub or PNG purifiers of water, and even to an extent of telling them, if you maybe don't like the taste of the chemical treatment, then you can even use ceramic filters. We have ceramic filters that our households can use to improve their water quality. And we encompass this also with social behavior change communication to make it sustainable. Why do you need to ensure that you make your water safe? So when they're taken through the water journey by these community health promoters, they're able to understand the need for continued consistency and use of these methods that they can make or improve their drinking water.
So Alex,
Yeah.
I have a question. So will you be around like after Sam's talk? So because we have
Yes, I am. Yes.
Okay.
Yes.
So,
Yes. I'm
so
very
we'll
much
continue.
around.
So I have more, like a lot of questions with you, but we have
Please.
to, we are a bit 5 minutes behind the schedule, 6 minutes
Okay.
now. So let me quickly introduce Professor Sam. So nice thing about Sam is like Sam is with us in Nepal as a Fulbright specialist. So he is professor of public health in University of Illinois in Chicago, and he's been giving us all different aspects of WAS and various technologies he's been implementing in Kenya. Actually, we came to know Alex through Sam himself. And he will be talking about a very interesting technology of disinfecting water using ozone on-site production, producing ozone on-site. And yeah, he very interesting fact is like he has been medical doctor, like he as a medical emergency medical interventionist He worked as medical interventionist for a long time, probably 30 years, if I remember correctly. And then he moved into research and now very active in water in the water sector. So over to you, Sam.
Well, thank you, Asim, for the introduction and for the opportunity to work with you and others at the institutes in Kathmandu, the RIB Institute and the Futung Research Institute. I'll be talking about, like you said, some new approaches to providing safe drinking water. And specifically, since this is a technologies of the future session, to talk about some technology that we're using. So this is called microplasma generated ozone for community scale water disinfection. As Alex and you have mentioned, Asim, that there are different approaches to disinfecting water at the point of use, meaning household water treatment. And you see WaterGuard, that's a commercial brand of sodium hypochlorite. There are ceramic filters. There's the approach that's referred to as SODIS or solar disinfection, putting bottles of water in the sun and letting ultraviolet light reduce the bacteria levels. These are inexpensive. It doesn't require big governmental infrastructure projects to make this possible, but they don't work so well. Adherence is always a challenge. The taste of chlorine is a barrier. The time it takes to do the ceramic filtration is a barrier. The same thing with the solar disinfection. So even though chlorination is maybe the best available approach, because of the adherence being low, the need for an ongoing steady supply chain of chlorine to homes, sometimes the costs of purchasing point of use treatment methods, these are all having a limited impact. Now, another approach to disinfecting water that's used in large centralized drinking water treatment systems in the US and in Europe, and also in wastewater disinfection, is ozone. And in the World Health Organization's most recent guidelines, they said that ozone is not recommended for household water treatment because it requires a reliable source of electricity to generate it, the complexity of generation and proper dosing in small applications, and the relatively high cost. So I'm going to make a case that ozone at the household level or at the community level is possible, and even without providing the electrical grid. And we're using an approach that uses plasma. So plasma is the fourth state of matter. There's solid, liquid, and gas, but plasma is a sort of a sea of ions. And the sun is a plasma, the stars are a plasma, and what you see here in this orange box is a microplasma, a chip that is in which in an aluminum block, a voltage is applied to a narrow channel about 250 microns in length, and air flows through it. And the oxygen in air, the molecular oxygen O2 in that electrical field is converted into O3 or ozone. So this requires very little electricity. We power it with a solar panel. Even if the electrical grid fails, the ozone generators can keep working. So the first time I encountered this microplasma ozone generator was at a conference where I met Professor Sung Jin Park, who is at the University of Illinois in Urbana-Champaign. It's the same university that I'm at, but at a different campus. And I was impressed with the concept, but I wanted to try it. So together with my colleague, Dr. Abhilasha Shrestha, we tested ozone on highly contaminated water that had sewage in it. And we measured what happens to the E. coli, what happens to the DNA of enterococci measured using the PCR process, and what happens to a virus, the coliphage, when treated with ozone. And what we found is that we reduced the levels of these wastewater indicators by about 2 log orders of magnitude. But in some cases, 2 orders of log orders of magnitude was the maximum that could be reduced based on the starting concentration. But it was impressive to see that within 30, 60, 90 minutes, we could reach significant decreases in the concentrations of E. coli. And what we did next was test it out in Kisumu County, Kenya, in collaboration with Alex and his organization, the Safe Water and AIDS Project. Here you see where Kenya is located, and within Kenya, Kisumu is in the western part of the country on the shores of Lake Victoria. And we used the ozone generator for point of use household treatment. You could see on the left side, here's a home with a solar panel on the roof, and it powers the ozone generator. The air comes into the ozone generator, and ozone enriched air comes out. It goes through this tubing and into a container of water. And as the ozone flows through the water, it produces bubbles, and that interface between the ozone and the water is the surface of that bubble. So over time, the ozone interacts with microbes in the water. And we found that at the household stored water level, we began with high levels of E. coli. This is water that's in the household brought from a local river or stream. The E. coli levels were high and the turbidity levels were high. And after 2 hours of ozonation, the E. coli levels went down to 11. And the turbidity stayed more or less the same, which isn't a surprise. The ozone is not there to reduce turbidity. So our conclusions from this were that this has potential to substantially reduce E. coli, but it did not produce safe drinking water. Another conclusion was that it's very difficult to do that in water this turbid. So if we could reduce the turbidity before treatment, we may see the production of safe drinking water with no detectable E. coli. And then another conclusion was that this was not a very efficient use of the ozone generator. One ozone generator is being used in one household for a couple of hours, and then the rest of the day it's not in use. So we thought that we should change all of these, and also to begin thinking about how an ozonation system could be financially self-sustaining. So what we did was to build a system called Solamaji, which means solar water in Swahili. This is our first site of community scale drinking water ozonation. And what we have here is essentially a drinking water treatment system. It has a flocculation stage, coagulation, flocculation. The water goes through carbon filters. And after that point, The turbidity is quite low, usually less than 1, almost always less than 2. And we have 4 ozone generators operating, 4 of those orange boxes. And in this ozonation tank, you could see bubbles of ozone that are moving through a 1,000-liter tank. And from there, there's a final filtration stage, and then water is dispensed at a kiosk. These are the components. You could see solar panels deployed on the left side, the ozone generators, and inside of this kiosk there are also the filters that reduce the turbidity. You could see on the right side here is the window at the kiosk. People come with their jerry cans and they purchase water, 20 liters of water for 5 shillings, which is about 4 Nepali rupees, or about 4 US cents. The water is also sold to water vendors, like you see on the left side, who then deliver it to their customers. So there are transactions taking place involving payment, and the payment we've found is sufficient to cover the operating costs of a community scale drinking water system. It does not cover the upfront costs of construction. We've since built another more modular system in Kenya. And then now we are working on what we call Solamaji 3. And again, at each version, we try to make it more efficient and simpler to use. So one thing that we're doing is using the ozone in line instead of having those the tubing and the bubbles in a tank. Ozone enters into the water treatment train through a Venturi system that pulls the ozone in. And we've been testing this in our laboratory in Chicago. Excuse me. Thank you. Sorry, I had to take a sip of water. So we spike water with E. coli. You could see that we start out with concentrations of about 50,000 per 100 milliliters. And as the water moves through that inline system, after 30 minutes, the water E. coli levels have gone down by about 4 log orders of magnitude. And this is, and we also control the turbidity. The turbidity here is usually 6 or 12, depending on the experiment. So fairly turbid water, 220 liters of it going from 50,000 E. coli to non-detectable in 30 minutes. So with that technology working, we're now building Solamaji 3. This is a school, a secondary school in a town called Kopere in Western Kenya. They don't have a safe drinking water supply. Adjacent to the school is it's called a dispensary, a medical clinic. They don't have a safe water supply. And there's a town of about 5,000 or 6,000 people, a very busy market area in the center of town, and they don't have a safe drinking water supply. No boreholes, and people get very contaminated drinking water from a river. So now we're building a 30,000 liter system, so scaling it up quite a bit from the earlier versions of Solamaji, where it was 1,000 or 2,000 liters being produced per day. You could see on the left, it's the almost identical inline system that we have in Chicago. at the university. The other thing that we're doing is, instead of pumping water from the river itself to the treatment system, we want to begin with water that's much less turbid, because it simplifies the treatment system. What we're doing is called riverbank filtration. Instead of taking water from the river itself, we're taking water from below the river. So the water, as it filters through the ground, it becomes much less turbid and the bacterial levels are decreased. This system will also have the capability of Internet of Things monitoring of system operation, sales of water, and aspects of water quality. This has a larger ozonation system. Instead of using one chip, this uses 5. So each chip is producing about 0.25 grams of ozone per minute. So this is 5 times the ozone production, but even though it's 30 times the volume of water, but because the water is much less turbid, this should be effective. And then from the treatment system, water goes by pipe to kiosks, where individuals can use a token, a digital token that is preloaded with enough money to buy 50 jerry cans of water. And there's no operator who is selling water at the kiosk. People can just swipe their token and get their water. So because there are not people working at these kiosks, it significantly lowers the costs, the operating costs, and it also significantly lowers the costs that we're not having to buy the alum, the flocculating agent. So this is not yet open. We hope to have a grand opening in maybe a month or two. But this is where we're heading now with our solar-powered drinking water ozonation system using microplasma technology. We still have a lot of questions to answer. How low can the cost come down? That's both for the capital costs of construction and also the operating costs. How can the systems be financed? So we work very closely with Alex's organization, the Safe Water and AIDS Project. And for the most part, the costs for the funds for building these systems have come from outside of Kenya. We got funding from the Bill and Melinda Gates Foundation for the first 2 systems. We have some funding from a prize that the team won from the United Arab Emirates, but we can't depend on that happening. So organizations like development banks are really going to be needed, because like Alex said, the government can't even pay for drilling a borehole. It's not going to be able to afford a system like this. We assume that providing safe drinking water reduces rates of childhood diarrhea and promotes health and wellness generally, but we need data to say that that's the case, and we don't have that data yet. We are also thinking about how to add sanitation facilities and even ozonating the waste when it's in a or when it's on its way to a digester. And then how can our partner NGO be supported when systems run into trouble? We had very strong winds that blew away a solar panel, or there's a mechanical issue. How to have enough funds not only to continue operating the system, but also the support system behind it. So these are technologies that are transferable. It's not magic that it's working in Kenya, and probably these systems could be deployed elsewhere. And we're interested in learning about partners overseas, elsewhere who might want to try it, and also funding mechanisms to support the construction of systems like that. I work for a university that owns the patent to that technology, to the microplasma technology. So I am disclosing an institutional conflict of interest. I personally have no financial interests in this technology. So this has been funded, like I said, the Gates Foundation, a local foundation in Chicago, the Portis Foundation, and the University of Illinois Chicago Center for Global Health, in partnership with SWAP in Kisumu, Shemgen Engineering, and the Eden Park Foundation in Champaign, Illinois. And I would be glad to take any questions that you may have. Thank you.
All right, thanks a lot, Sam. So very interesting question that we have to think about from Om Vishnu. So his question is, if implemented in Nepal, what challenges do you see in using your technology in Nepal's rural areas, and how can solar power can be made more sustainable?
Okay, very good question. So the cost of solar panels keeps getting lower and lower. One challenge would be, is there clear weather enough of the year to power a system like this using solar panels? If there's a monsoon and it's very cloudy for weeks at a time, that may make the generation of electricity using solar power challenging. Another challenge I would anticipate is, are people in those areas of Nepal used to paying for water? To keep this system operating, we sell water. The local governments in Kenya set the price, so it's a pretty low price. But if people in Western Nepal typically just go to a river and collect water, they may not want to buy water. So that would be a problem I would anticipate. This isn't the solution to every drinking water supply problem. If there's good groundwater and the groundwater, is the withdrawals do not exceed the recharge rate, maybe groundwater is a better solution. It would depend on the circumstances. Clearly, one of our secrets of success is our partnership with Alex and the Safe Water and AIDS Project, a community-based organization or a local governmental organization that really wants to take the initiative and make something like this happen is critical. We had to go through many different kinds of permitting processes, hydrologic surveys, paying for the Bureau of Standards in Kenya to test our water and confirm that it meets national drinking water standards. So, partners are needed who are willing to do all of the administrative and managerial aspects. We also couldn't have built this without our civil engineer partner, the Shemgen Engineering. And they're the ones who actually dig the trenches and put up the solar arrays and the the battery storage systems. So an engineer is needed. So those are some of the anticipated challenges. One challenge that I would not anticipate, unlike chlorinated water, is people like the taste of the ozonated water, and that's not always the case with chlorinated water. Another thing that people might like is having a solar array may bring electricity to a community that doesn't have it. And in addition to being able to power a drinking water treatment system, it could provide electricity to a school, or people could charge their mobile phones, There are other things that may incline people to support this type of system.
So there's, I think, something related to this. So in the trekking regions of Nepal, so there are some ozonated water, kiosks that, it's mostly for tourists. So the locals usually don't do that, but it's because it's water bottle filling stations. And they don't use this low cost technologies, but they have been somehow been operating, and it's for free. So I don't know the mechanism, just for the information here.
Well, I'm surprised to hear that because, the conventional methods for producing ozone require a lot of electricity, and they don't work well with humid air. And really, they work best with pure oxygen. So if pure dehumidified oxygen is fed into in a strong electric field, it'll produce ozone. So I'm curious about how that's being done in the trekking region.
So it's not in all the trekking region, but it's in the towns
Uh-huh.
of the most popular tourist destinations. So I have
Interesting.
seen that, yes. So the next question is from Ashish. He says, "Is ozone poisonous?" It's sometimes called. And if so, what is the safest way to use it for water purification?
Okay, excellent question. So ozone is a double-edged sword in a couple of ways. One is, we know that ozone is good because it is the ozone layer, and that protects us all from UV radiation, solar UV radiation. But at the same time, I would imagine that on a very hot, very sunny day in Kathmandu, the ozone levels are, close to the ground, are elevated. And it probably is causing some cough and wheezing and problems. And just like in any city in the world that's got a lot of motor vehicles on a hot day. So that ozone, tropospheric ozone, close to the ground ozone, is harmful. Ozone can cause irritation of the eyes, nose, throat. So it would not be safe to use that indoors. But in a well-ventilated space, or in our case, outdoors, any ozone that leaves the system is quickly diluted. The other thing is that ozone is an extremely potent oxidant, but as soon as it oxidizes something, it is reduced back to O2. So it's highly reactive. So if it contacts any material, pretty much, not too many things are inert to ozone. It will be reduced back to oxygen. So when we did our study of providing families with ozone generators and solar panels for point of use, we asked people questions periodically. Do you smell anything? Does it cause irritation? Is anybody having a cough? And nobody experienced any problems. But the way we're doing it now at a centralized community site, members of the public are not nearby. They're at the kiosk getting their water, so they have no ozone exposure.
Right. So the next question is, how does ozone-based water treatment compare with other purification methods in terms of cost and effectiveness?
Well, right, at a large scale, like at a large American or European drinking water treatment system, the costs are really dependent on the costs of electricity. I think they are comparable to chlorination. The systems we're building are, we don't have, we're not at full scale. We don't have a large volume of projects. So there are a lot of inefficiencies that or we don't benefit from the efficiencies of having lots of construction going on all the time. So chlorination is definitely, at a small scale, much less expensive. Chlorine also has the advantage of it leaves a residual in the water. So the free chlorine residual will prevent the water from getting recontaminated. That's not the case with ozone. There's no residual. So safe water storage is very important. If ozonated water goes into a home and it's stored in a container and children are putting their dirty hands in it, the water will get recontaminated. But if chlorinated water were to have kids stick their hands in it, if there's enough chlorine in the water, it'll prevent bacterial growth.
Right. So the next question seems like from Suraj, who seems like from a chemist. So he asks, will ozone-based treatment react with manganese and iron impurities in water?
Yes, it will. It will oxidize metals, and we've seen that here in Nepal. We saw some of our equipment turn rust-colored because the iron in the water was oxidized. Another thing that's important is to know the geology and the chemistry of the water. Because if there is bromine, naturally occurring bromine in the water, of groundwater, it'll be oxidized to bromate, which also has adverse health effects. So, by no means am I trying to promote ozone as this, magic bullet. It has no negative effects, only positive. It works everywhere. Not at all. There's a lot that, a lot of limitations. On the other hand, there are methods for removing metals from water. If that's being done because of high iron content, then that would make it much less of a problem to ozonate that water.
Right. So there seems to be no more questions. So let's keep the discussions going. So I will try to relate what you just said while you just gave a presentation to what Alex, I was asking with Alex. So in one of your slides, you said that your water was turbid, like a bit turbid, and then you did the ozonation. So the turbidity was about 6 to 12 NTU, and then you saw that the E. coli got reduced, right? So that is not according to WHO standard because it has to be less than 5, right? So, but you are providing water that is relatively free of E. coli, right? So, what do you say about it? Like connecting with what I asked to Alex.
Well, we are compliant with the WHO turbidity standards as well, because at the very first in the very first place, where we provided point of use drinking water ozonation systems to families, they didn't do anything to reduce the turbidity of their water. But we do initially through coagulation, flocculation, and filtration. And now the intake water is very low turbidity because of the riverbank filtration. We're not, the river itself may have 50 or 100 or 200 NTU turbidity measurement. But once the water is withdrawn from underneath the river, the turbidity is around 1, something like that. So we aren't compromising of providing, E. coli free water at the expense of meeting the turbidity standard. I mean, that said, turbidity doesn't make people ill. So, if I had a choice of, providing people with bacteria-free water that's slightly turbid, or crystal clear water, was, turbidity of 0.01, but there's bacteria, I would say let's use the bacteria-free water. But I don't think it has to be an either or choice.
Okay, so let's keep the discussions going. So I want to ask this question to Alex. You did briefly mention about the gold mines, the pollutions due to gold mines in Kenya, right? So, what do you think about? I mean, what should the world do? Because I think it's a serious problem. You mentioned that, and you highlight your experience regarding the pollutions due to the gold mining and the fact that it is somewhat neglected problem. People do not do a lot of research, or do or may or may not do. So can you discuss a bit about the pollutions due to gold mines, and what kind of research needs are there, or analytical needs are there? And I hope Heidi is listening too, and I'm listening, and more people involved in technology research are listening to you as well.
Yeah, so thank you so much again. This has been a gray area of research, trying to find out the extent of water pollution in gold mine areas. What we have recently done was to detect levels of mercury that is used in the gold mining, and we are seeing this relatively high pollution, especially in those gold mine areas. And when they drain this water, again, it pollutes the water bodies. We are currently working with the University of Illinois at Chicago, Professor Sam, and even our own research institute, that's Jaramogi Oginga Odinga University of Science and Technology, just to find out what are some of the hazards, especially in relation to water quality, in relations to human health, but it's a gray area that many people have not really done a lot of researches in Kenya. And that's the reason why we are trying to come up with ideas. How can we research on what goes around in those gold mine areas? There's problem of noise pollution, the dust, and that also contributes also to health hazards of the people living within the these gold mine areas. I know as far as water is concerned, we want to look at those levels of metals, heavy metals in water. But I know recently also we've had disasters also, other than just looking at water, but we have also encountered serious disaster occurring, especially in these gold mine areas because of the collapse, people trapped in those mines. And we've had a situation where in one part of the country, they had to shut those places because of mortality that were experienced. Yeah, but it's something we are considering doing a lot of research moving forward.
So what are the main pollutants in these like gold mine areas? Like heavy metals you mentioned, but what specific heavy metals, or if you have,
Yeah, I
because
think one of the
sorry.
yeah, so one of the heavy metals that we experience is the mercury, which again is used for the process. But again, last year we were trying to visit some of these areas just to find what kind of metals are we likely to encounter. And we are just in the process of doing the testing in collaboration with the University of Illinois at Chicago. And soon we are expecting to find what are the results from the samples that were taken to do further research.
Right. Thank you,
Yeah.
Alex. So the question now is for Hedy. So the question is like very technical question. So there is this issue of like let's say focusing on mercury pollution in Kenya. So in terms of technology, let's say optics, what could be a potential solution or to see the exposure, test the water, for example?
Yeah. I assume, because this was still a question I have for Alex, I assume you want an online monitoring system such that you don't have to go for manual sampling, but that you have a kind of sensor that gives a warning to the appropriate people once you go above a certain threshold. I assume that is what you want, correct?
Alex?
Yes, very, very, very correct. And I think this is the new research area we are exploring together with the University of Illinois,
Yeah.
coming up with even remote sensors for instance in issues of pollution. And I think that's the way to go, trying to find when situations where there are excessive limits, how can we arrest that situation?
Yeah. Okay, yes, so with respect to technology, I think we can keep it quite simple here, and even absorption measurements would already do the job. I know there is an Austrian company, Escan, but they were recently hired by a larger American company, but I forgot the name. And I know they have sensors for detecting metals, and mercury is one of them, because it's not only in Kenya, but in other countries, other developing countries as well. Now, the cost of these probes, because it's a probe that goes together with a controller, is still quite expensive. It is above, I think, $3,000 with the controller and the probe itself. So I think the challenge will be now to look to much cheaper technologies where LEDs and photodiodes with some filters in front. If you know the real targeted lines or wavelengths at which you want to look, you don't need a spectroscopic system, where you can just look at targeted wavelengths and capture those signals specifically then for a few metals you're interested in. So I think that is the current challenge if you want to bring it to make it cheaper, such that it can be widely deployed.
So what technology is it? Is it like spectrum, like absorption?
Yeah, they do absorption spectroscopy at this stage, but that's what I say. Of course, this is a quite generic probe they develop, and that's the reason why you have this high cost, because the spectrometer on itself is quite sensitive, and it's too expensive for that type of sensor. So I believe that it would be a good idea first to identify for a set of heavy metals what are the absorption lines, and then to look for some LEDs or other cheap sources at those specific wavelengths, and then look together with some algorithms or AI even, get out what you need to know and give a warning. Of course, there also need to be installed some electronics or some communication with a solar panel, as we also heard in the previous presentation. So, but according to me, there exists already literature. There are even commercial products, but for developing countries, they are too expensive and they are too generic. So, better to focus on a few aspects and while reducing the cost. I think that
So
should be done.
would fluorescence like binding kind of technology, does there is there such technology where there is like some binding chemistry?
Yes, but then of course you have again a lot of chemistry, yeah? And maybe people do not. It's not that easy to use always. Then indeed you have some some molecules that bind to some of the metals, and as such can detect. Now it all depends, of course, what is your limit of detection you want to reach. now I know with the sensor of scan, they have quite a long interaction length and they can go quite sensitive. I don't know by heart what are the detection limits they can get, but it's going quite low. Of course, if you would have a few metal molecules in liters of water, yeah, to find those, that might be more difficult, of course, and then you need maybe some dedicated filtering or indeed some processing with functionalized molecules, but that makes again things more complicated. I personally think, if I hear what Alex says, that to that level of contamination, that even a simple absorption measurements could do the job. That's my gut feeling.
So this morning, we were discussing with Sam, and he mentioned, so the typical or the clinical level has to be about 5 of some units, but the in the children it was more than 200. So I think even if we have detection limit of 5 of that unit, I mean, should be very helpful to
Yeah,
communities. Yeah.
indeed.
Right. So the next question again goes to Alex. So we heard something about microplastic detection here, right? So do you have do you even worry about microplastic yet in your country, or you have started thinking about this issue?
Yeah, so I know microplastics is one thing also as a country. We have witnessed, especially like about 3 years ago, the country banned use of plastic papers for wrapping, and this was attributed to, again, issues of pollution, including water bodies. But then we still have a problem, especially in terms of packaging of bottled water. There's quite a lot of bottling of water using plastic materials. But again, I believe that there still need more studies to look at the extent of microplastics pollution. But we have come a long way, where I find most products available in the market these days are made of plastics, and that has been quite a worrying trend. Yeah, so it's good from the presentation I saw from Heidi, some of the technologies that are now being used to measure levels of the microplastics. Over.
Right. So if there is any question from the audience, so we'd like to have a question from the audience. So please type in your, or if you want to speak, just on mask yourself, unmute yourself, and you can ask. So in the meantime, while you are thinking of the question, so let me ask. So we are here, community people engaged with the community, some researchers, public health, experts here. So the question is, in general, it's a bit general. How can we collaborate? How can we keep this conversation going and keep on engaged? And just some suggestions for me to carry on this project. I mean, just some comments, questions, suggestions from the speakers.
One of the possibilities, I think, is definitely to exchange researchers. And even it should not be for very long, because of course that goes together with a certain cost. But even for short visits, I think it might be very helpful to have exchanges in both directions, that people from developing countries can better understand technologies and commercial products that are available, while on the other side, we can more learn the real problems in the field to see how those can be solved. I think that's definitely an option, exchanging of research. It's definitely also of added value for the persons themselves. to have some international experience. That's one thing. And secondly, I think there are funding opportunities through international organizations to collaborate, not only within the continent, but cross-continent. And for that, maybe some specific brainstorm sessions can be organized to define some projects.
All right. So coming back to that, so I remember when I was in a committee, the educational committee back in Erasmus times. So we discussed quite a lot about mobility, right? So I think so from Futung and from the organizers, Reeb and our organizers, we are very much open to host any researchers who might want to come from your institute to here. From here to there could be, we need probably some more funding, but from there to here, they can come here as a tourist for a bit and then spend some time here and,
Yeah. There I have a question, Ashim, but maybe
Sure.
we should not discuss it here with all the people involved to not to take their time. But are you linked to a university? Because there are special programs for university students out of Europe to request for funding, just as there is the Erasmus scheme
Yes.
for mobility in Europe, but it also exists out of Europe. But it's not with a research institute, but it's with a university.
Oh, we have connections. So tomorrow we're gonna have a very big meeting about a new university we're trying to organize. So
Yes.
I'll be in touch with you.
Yes, because that's definitely an option. And so the flight is paid for those researchers as well as their stay here. So that's very interesting.
All right. So Alex, so how can we continue our conversation and collaboration? Any suggestions that comes up in your mind?
Okay, yeah, so very good question. Maybe I could give you an experience we have in Kenya. In Kenya, we have networks at the counties and even at the national level. And these networks would bring like-minded organizations, and it will include both private, public sector, including academia. And so these networks would organize of late after the COVID-19, people have decided to do most things virtually. So we have series of webinars that are hosted, and it's a platform that people would share knowledge, experience. Of course, we also have hybrid where people can attend in person, and if you're not able to attend, then you can be able to join virtually. And we have seen people sharing in this platform a lot of knowledge, experiences when it comes to WASH-related issues.
Right.
Over.
Yeah, thanks a lot. I think we will. I will write to you, and of course, Eddie and Sam and everybody who is participating here can write to each other and continue the collaboration. So I will ask Sam to answer the same question, probably like try to summarize what happened today briefly, and then close the whole workshop. I call online workshop.
Okay, well, thank you. I think it's been a very exciting workshop. Fantastic speakers. I'm just thinking about each talk and the different perspectives that everybody brought. So Purnika was talking about variability by season and site in measures of water quality. And I think that brings up really important issues that, we're working with very dynamic systems, and that we should all be thinking broadly and obviously recognizing how the real world challenges we're trying to address are far more complicated than what we are doing in laboratories. Professor Geary talked about the microbiopad and the smartphone microscope and methods for measuring microplastics in water samples. These are really wonderful ways of addressing problems in low and middle income countries without having to purchase expensive equipment. That, you know, if there are simple tests that the pad or that the cell phone camera can do, it really extends the range of impact of the technologies. So I think that's very beneficial, and it was great to hear about that. And what I'm sorry, Professor Heidi, I'm not sure if I'm going to pronounce your last name right. Otavare,
Perfect.
is that right?
Yes.
Okay. So, you know, there are laboratories around the world, advanced physics laboratories that are doing many types of photonics work and computing work and microfluidics work, but taking that and applying it so that it could be used in low-income countries is really important. And it's great that there's a pipeline that brings advanced methods to be applied in real world settings where substances can be analyzed without expensive laboratory equipment, and trying to think about ways of doing impactful work, especially on emerging pollutants like microplastics. I think that that's been very fascinating. I think Alex brought a context to the issue where, you know, most of the people presenting here are scientists, laboratory researchers, and Alex brought a community organization perspective. And he also really highlighted some of the challenges that exist in bringing safe drinking water to all. Kenya, it sounds like, has very good written policies, but the implementation is lagging, the financing is available, the lack of integration between local government, county government, national government, and then even within a level, different ministries or departments are approaching complex problems from a single perspective. So to prevent cholera outbreaks and the continuing challenges of inadequate wash, cross-sectoral research is needed and cross-sectoral cooperation is needed, so that solutions can be implemented. And another thing that Alex mentioned that I think really ties back to some of the other speakers' comments is that wash indicators are often lacking, and data about water quality is lacking, and that deploying sensors that in real time could perhaps send an alarm that there's a major water quality problem occurring at a certain location, that could be very useful. So that's my quick summary, trying to distill what was said and to highlight how things that different speakers said are related to each other. Thank you, Ashim, for allowing all of us to participate in this conference.
Yes, thanks a lot, everyone, for joining in. So we are 6 minutes behind the schedule, but still, we still have 25 people. Yeah, but yeah, that's really great. It was a long, without a break, by the way. So thanks a lot for being here, and we hope to continue the conversation in the coming webinars. And I think we will slowly move on to making the collaborations, developing, finding the solutions, collaborating, and that's will be our focus in coming webinars. So thank you very much for joining in, and hope to be engaged with everybody in the coming days. Thanks a lot. Thanks a lot.
Thank you,
Ashim.
Ashim, and thank you, everyone.
Bye-bye.
Thanks,
Bye-bye.
everybody. Bye-bye.
Mhm.